Continuing with our fascinating discussion of the respiratory system. No, wait, the muscular system. We have to look even deeper than what we had learned before. We learned that the muscle belly is made up of fascicles, which are made up of muscle fibers. Now we have to dive down inside to see that the majority of the interior of the muscle fiber is occupied by a specialized organelle of contraction called the myofibril. The myofibril is the heart of muscle. No, that's not a good way. Can I say it a different way? The myofibril is the essence of meat. Meaty tissue is meaty because it is mostly protein. And really what makes a muscle cell mostly protein are these, the myofibrils. They occupy something like 90% of the interior of a skeletal muscle cell. So, although the muscle cell has all of the other parts that our sort of generic cell from chapter 3 had, think rough ER, smooth ER, mitochondria, it has all of that stuff. It's not a round cell, it's not a squishy, watery cell. Muscle cells are mostly protein, and it's because muscle cells are mostly myofibrils. So when we add that all up, the muscle belly is mostly protein. And we know that if we let ourselves go, there might be some deposition of fat, some marbling that we would find on the inside here. And it's still true that it's mostly protein. And in young and lean and healthy people, of course, a muscle has very little fat at all. So here we see the source of all of that protein. The myofibril, actin, myofilaments, and myosin, myofilaments. If we pull one of those little sections, one of these little stripy bits out, we can see that the actin is actually a part of this blue and purple structure called the thin filament. And that the myosin, shown in green, is a part of what we call the thick filament. This structure is called a sarcomere, and it is the smallest functional unit of contraction within any muscle cell. It just so happens, because of the string cheese packaging arrangement and the parallel alignment of our skeletal muscle cells, when we look at it under a slide, all of the sarcomeres line up, as if many, many trains pulled into the station, but the front car lined up perfectly. These are all the engines, and these are all the coal cars, and this is the first car and the second car. If you look really closely, you can see this is one myofibril made of many, many sarcomeres in a row. Sarcomere, 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 sarcomere. Here is another myofibril, and a third and a fourth, all packaged inside of this muscle cell, which in a very confusing way is called a muscle fiber. So listen to this. The myofibrils are the organelles of contraction, and they're composed of myofilaments, the thin and thick, actin and myosin, and they all exist within a skeletal muscle cell called a muscle fiber. So this is some heavy lifting, cognitive heavy lifting here, people. Flashcards, review, talk about it. You've got to get other people involved. You can't do this by yourself. Please, please reach out to each other. Start forming study groups if you have yet to do so, or get into the Academic Support Center. When my muscle contracts, it's because my nerve tells it to. I say I want to move my hand. The nerve comes down, tells the muscle to contract. All of the sarcomeres contract together, so this whole muscle cell shortens. And ultimately, at the heart of that process is this amazing walking of the myosin heads along the actin filament. And if you thought this was detailed, just wait for the next part. Look at this. We have to understand all of these parts, and then all of these processes that allow the muscle to shorten. Let's watch this video to really see how it works. Oh! The video does not work. Un momento, por favor.
Nerve impulses also.
So when I want to move my arm, it's amazing, but I really just have to think about it. And a neural impulse is sent down out of my spinal cord and into this long motor neuron. Although it looks like the electrical activity sweeps directly from the neuron to the muscle cell, remember that it actually uses chemicals called neurotransmitters to sort of spritz out, in this case, acetylcholine, an excitatory neurotransmitter that's going to bind to tiny keyholes, tiny receptors on the muscle cell surface. Here we see the process in detail, and let me remind you, as complicated as this is, you're getting 2 for the price of 1. If you can learn how a neuron and a muscle cell communicate, you'll really be well on your way to learning how neurons communicate in the upcoming neurology unit. So here we see the action potential sweeping down the neuron. It causes these little bubbles called vesicles to dump out their product. Remember exocytosis? Well, that's what we're going to see right here. Calcium causes these vesicles to fuse with the plasma membrane, spilling the neurotransmitter out of the cell. The shape of this chemical, acetylcholine, is just perfect to fit into these little chemical receptors. These are called nicotinic receptors, and they are a form of acetylcholine receptor. Think of this as the key, and this as the keyhole, right? This is the signal, this is the antenna, however it works for you. Chemically, this is just spritzed across a tiny area. So these chemicals cross the gap in almost no time at all, and then bind tightly to the little green molecules. These gated ion channels respond to chemical signals. Ligand-gated cation channels means chemically activated doorways for ions, in this case a positive ion, sodium. This arrangement in the frequency that these doors open is going to tell us how frequently this muscle cell contracts. Once the acetylcholine binds to the neurotransmitter receptor, the acetylcholine receptor, it allows for the exchange of ions. Potassium flows out, and sodium rushes in, and it causes something like a tiny spark within the cell. Even though these ions are both positive, we'll learn later on exactly how this works, but the exchange, that flux of ions, is going to cause the muscle cell to get really sparky and then send an electrical activity down and into the cell. We need enzymes to chop up those neurotransmitters and get them out of the way. And in some cases, there are specialized channels or endocytosis that suck the neurotransmitter out of the synaptic cleft and back into the presynaptic neuron. Here come the enzymes, acetylcholinesterase, which breaks the acetylcholine apart into its components, which can then be re-imported and recycled to make more of the neurotransmitter for the next firing. It's really amazing. In the meantime, they've cleared all of the neurotransmitter out of the synapse, and the muscle cell will now go into a state of relaxation. Looking a little bit more deeply into that process, if we could use little electrical detectors plugged into the cell and sort of listening to see what the charge does along the cell membrane, we would end up generating a graph that looks like this. Here's the electrical activity before the muscle cell fires. Here's the electrical activity as the muscle cell is being stimulated until it reaches this very important point, threshold. Threshold means there's no turning back. We're go for launch, and the electrical activity causes a significant flux up into positive territory, and then very quickly back into negative territory, and then it resets for the next firing. This peak is called an action potential. And the different parts of the action potential wave are what we see here. Resting membrane potential. Here we see it right here. Resting potential. What they call polarized is usually right down here at about negative 85. In many neurons, it's negative 70. And once the sodium begins to leak into the cell, it carries with it a positive charge, bringing the cell closer to threshold. Once that electrical activity hits this point, the electrical vibration in something like an electric garage door opener, the electrical activity causes a physical change. So when I push my garage door opener, the electrical wave turns on the door and it opens. In the same way, at the super small cellular level, threshold is that electrical frequency. It causes all of these voltage-gated ion channels to open, and suddenly sodium rushes into the cell. At the top of the curve, it opens different doorways for potassium, which rushes out of the cell and takes with it the positive charge. This downward deflection is called repolarization. At the bottom of the curve, sodium and potassium are in the wrong position. Sodium, having left the cell, and potassium, which is now on the inside of the cell, causes this curve to be in hyperpolarized territory until the sodium-potassium pump brings it back to resting membrane potential. Now, it's kind of ridiculous, but you have to know all of that. And so by practicing drawing this graph, and by looking through all of these slides in a painstaking fashion, you can understand how the movement of these ions will sweep down the neuron all the way to the end. The synaptic knob, or presynaptic terminal, will then release the chemicals, exciting the next cell in the arrangement. Now, to help you, there are some much better videos than this one that I have hyperlinked on Blackboard for you to link to YouTube and watch those videos. Ride the pause button, work your way through this material. It is certainly very challenging. The next step of this process that I'd like to talk about is how the electrical activity, once it's started on the cell of the muscle, dives down into the interior through a specialized tube called a T-tubule, a transverse tubule. T-tubules carry the electrical activity down into the cell, and then they cause these yellow organelles to spill their guts. They dump out all of this calcium, which starts the next process, allowing the thick and thin filaments to hook together. This process has to do with the electrical activity opening voltage-gated channels, voltage-gated calcium channels on this yellow structure we call the sarcoplasmic reticulum. The sarcoplasmic reticulum dumps the calcium into the sarcomere, where it causes these thin and thick filaments to begin to stick together. And if there's enough energy in the form of ATP, the green ones will start walking down the purple ones, sort of in the same way that a caterpillar moves, pulling the thin filaments and shortening the sarcomere. Who are these important proteins? Well, on the thick filament, we see the myosin protein. In little pairs like golf club heads, they're going to be the feet of the caterpillar. The actin myofilament is made of 3 unique proteins: actin, troponin in red, and tropomyosin, which is this long white ribbon. Once calcium binds to the troponin, it pulls the tropomyosin rope out of the way, exposing all of these yellow bits, the active sites, which are the only part that the myosin can grab onto. So this is really at the heart of the matter. Our ability to contract is dependent on this tiny temporary chemical bond called a cross bridge. And it doesn't happen until the rope moves, and that doesn't happen until calcium enters the picture, and that doesn't happen until calcium is released from the sarcoplasmic reticulum, and that doesn't happen until the electrical activity comes down the T-tubule. So I highly recommend cutting this apart, making flashcards, putting out post-its. There's probably 17 or 18 unique steps depending on how much detail you go into, and you should be prepared to tell me each step in order which proteins are involved, which chemicals or electricity trigger the process. This is why it's called college level anatomy and physiology. You'll feel better for it. And honestly, if you go into physical medicine, or if you work with patients who have neuromuscular disorders like cerebral palsy or muscular dystrophy, it will really help you to understand what's going on in your patients' bodies by working through this. So maybe that'll help to motivate you to do the hard work here, to work your way through all of these steps. So make a flashcard for each of these steps. Now, what we see is this event is really a one-time thing. Firing of the action potential on the neuron is coming down in a quick little spark, just one little flick. That stimulus causes many, many of these cycles to happen. One little spark, which we call a twitch, results in many, many individual loops of this cycle. So this process is happening so fast, it's very difficult even to appreciate it on a contraction and relaxation sort of time scale. Muscle relaxation is dependent on the neurotransmitter being cleared from that gap and then the calcium being sucked back into the muscle cell. So if you really want to work through it, you need to know all of this stuff. This is actually an excellent activity. Make a flashcard for each of these phases. Here's the cross bridge cycle. Here's what happens in relaxation. How does the neural impulse get from the neuron onto and then down into the muscle cell to cause the troponin tropomyosin complex to change? Okay, so in all reality, you don't want to spend too much time reading this. This is a good summary, but as quickly as you can, you need to be actively sorting each of these into its own little compartment. Watch all of the videos that I've posted for you on Blackboard. Make flashcards, draw pictures, use Play-Doh, sculpt it, sing it, rap it, dance it, whatever. You got to be active. And the very best way of learning all of this, I think, is to start with the videos until you can really see it in your own mind, and then do what you have to do so that you can transmit that information onto paper on test day.